Today, I would like to talk about the metabolome analysis of plasma using the kit in a large cohort study in Japan and its application to gynecological cancer biomarker discovery. Although there are various methods to analyze human phenotypes, such as genome, transcriptome, and proteome, metabolome is the most downstream of the central dogma and is considered to best reflect human phenotypes. Therefore, metabolome analysis is considered to be useful in searching for biomarkers for disease prevention and diagnosis. The map of metabolic pathway of the organism is shown here. There are numerous metabolites in the human body, and their properties are very diverse, ranging from hydrophilic and hydrophobic. Especially in our large-scale cohort study, it is necessary to measure a large number of metabolites with high throughput and accuracy. Tohoku University, Tohoku Medical Megabank Organization, ToMMo, was founded to establish an advanced medical system to foster the reconstruction from the Great East Japan earthquake. The organization has been developing a biobank that combines medical and genome information during the process of rebuilding the community medical system and supporting health and welfare in the Tohoku area. The Advanced Research Center for Innovation in Next Generation Medicine, Eiji, was established in 2017 as the hub for next generation medicine to bring together the university's medicine as a university's preeminent research capabilities in fields such as information science, basic life science, and genomic medicine as a core subject. Eiji has established a clinical biobank where biological samples of patients with various diseases, mainly from Tohoku University Hospital, are accumulated, and we are conducting omics analysis of these patients' derived samples. Previous metabolome analysis in ToMMo using mass spectrometry is shown here. We used to perform metabolome analysis using global metabolomics and GCMS, but since these results are relative values, it's very difficult to compare batch to batch and between facilities. Recently, however, biocrates has developed a target metabolomics kit, and we have used it to perform metabolome analysis on a number of plasma samples. The features of the absolute IDQP 180 kit and the MXP Quant 500 kit are shown. These kits are very useful for our large cohort studies because they can quantify a large number of metabolites in a very high throughput manner. Furthermore, the amount of sample used is very small, and normalization between batches is very easy. This slide shows the analysis flow in ToMMo and Eiji. We are using commercially available plasma as our own globally quality control ZQC. And we are applying ZQC to 4 wells per plate. We have performed many plate-to-plate normalizations using the quantitative values of these ZQC samples and have obtained very stable results. In addition, NIST plasma is added to 1 well per plate to check the quality of the data. We have measured approximately 10,000 plasma samples from the ToMMo cohort and more than 1,000 plasma samples from the Eiji Clinical Biobank for cancer, diabetes, and cardiovascular disease. Other samples such as serum, urine, bile acid, feces, and exosome have also been measured. NIST is internationally used human reference plasma. And many quantitative values of metabolites measured by various methods have been reported. We used this reference plasma to check the accuracy of SP-Quant 500 kit. This data compares the quantitative values of representative metabolites using the MXP-Quant 500 kit with those measured by NMR. The graph on the right shows the ratio of respective quantitative values, and most of the metabolites have well-correlated quantitative values, while the 6 metabolites shown in orange have more than twofold differences, indicating differences depending on the quantification method. We have published the quantitative values of metabolites in plasma samples of the ToMMo cohorts obtained using the kit in the Japanese multi-omics reference panel, ToMMo, shown here. This database can be accessed via the internet to obtain metabolite information as well as genomic, transcriptomic, proteomic, metagenomic, and pharmacogenomic information. The database of metabolites looks like this picture. The metabolite common name, ID, number of samples, quantitative values, and links to other database displayed in this way. You can see a histogram like this by clicking on the ID or name of each metabolite. Some metabolites change in relation to age, gender, and BMI. For example, there is a tendency for CE25 to correlate with age, and for creatinine to correlate with gender. In addition, the quantitative values of NIST plasma and ZQC measured by us are also published in this database. So these quantitative values can be used. And this year's update also allows metabolite structures to be displayed. Here is a graph that visualizes the quantitative values of the ToMMo cohort samples. The left shows the differences by gender, and the right shows the differences in metabolite profiles for each individual. In the graph on the left, the blue line represents males. The red line represents females, and the black line represents the median. The graph clearly shows that triglycerides are higher in men, while sphingolipids are higher in women. We call this graph the metabolo index. Index can be utilized for creating the personal health care program and searching the predictive biomarkers. Also, in this graph, men are on the left and women are on the right, with darker colored lines indicating higher BMI, and lighter colors line lower BMI people. In both cases, TC was found to be higher in those with higher BMI, with PC, including PUFA, was higher in those with lower BMI. In the next section, I will talk about the results of comparing metabolite profiles for ToMMo cohort samples and cancer samples in the Indian clinical biobank. Cancer has been the leading cause of death in Japan in recent years. With 9.6 million people lost worldwide in 2018. The number of cancer patients is increasing every year, and it is necessary. It is a very important disease worldwide. Cancer cells are known to reprogram their intracellular metabolism, and in general, the Warburg effect causes the glycolytic system to become dominant. It has been reported that a variety of characteristic metabolome pathway changes and metabolite fractions occur accordingly. Therefore, metabolome analysis is useful for identifying biomarkers for diagnosis and prevention of cancers. Initially, we analyzed several types of cancers using the P180 kit and compared their metabolic profiles with those of the ToMMo cohort samples. This slide shows the result of PCA and S-plot. The analysis showed that there were differences in metabolite profiles between cancer and cohort samples, especially tryptophan, histidine, leucine, and valine were decreased while glutamine was increased in cancer patients. There was also a decrease in lysophosphatidyl suggesting increase in LPA production associated with the inflammatory response. This slide shows the metabolites with particularly significant differences. Histidine, valine, and LPCs show large changes. Furthermore, increase in kynurenine was observed while tryptophan and serotonin decreased. They were involved in the indole metabolic pathway, suggesting that activation of the kynurenine pathway is involved in cancer progression. So this is the result of comparing the metabolic profiles of 7 ovarian cancer patients before and after cancer chemotherapy. All patients receiving TC therapy, paclitaxel plus carboplatin. The red line is the mean value of the cohort samples. Patients numbered 1 to 6 have a good prognosis with metabolic levels recovering to the same level as healthy individuals after treatment with chemotherapy. Patient number 7, on the other hand, has a different cancer subtype and a worse prognosis. It was found that metabolic profile levels were negatively correlated with other patients. Next, we then compared ovarian cancer samples with those of the cohort samples. A targeted metabolome analysis was performed on 80 ovarian cancer patients and compared metabolome profile with 80 ToMMo cohort samples that matched age, height, weight, and BMI. PCA results showed slight separation of the metabolic profiles of ovarian cancer patients and ToMMo cohorts. Whereas OPLSDA results showed strong separation, suggesting a characteristic metabolome profile for each group. Compared with the ToMMo cohort, 77 metabolites were significantly increased, and 140 metabolites were significantly decreased in ovarian cancer patients. Here is a heat map of metabolites that showed significant change. As in previous results, there is a significant decrease in histidine, serotonin, and PC, and a significant increase in TZs. In particular, the decrease in PCs containing unsaturated fatty acid was found to be significant in ovarian cancer. The results so far suggest that the kynurenine pathway plays an important role in cancer progression. Therefore, we confirmed the relationship between the ratio of kynurenine to tryptophan and the prognosis of these ovarian cancer patients. When the kynurenine tryptophan ratio was compared between the CRPR and SDPD groups for efficacy of first-line chemotherapy, there was a trend toward higher ratios in the SDPD groups, but no significant differences were observed. However, when compared by patient prognosis, the worse the patient's prognosis, the significantly higher to ratio of kynurenine and tryptophan. The kynurenine pathway is enhanced in ovarian cancer patients with high indole amine 2,3 dioxygenase, IDO activity, and may be involved in the deterioration of ovarian cancer by suppressing tumor immunity. This slide shows the mechanism of kynurenine and cancer progression. The kynurenine to tryptophan ratio reflects IDO activity, and the higher this ratio, the more kynurenine production is enhanced. Kynurenine acts as a ligand for the allyl hydrocarbon receptor, AHR, regulates the transcription of various genes, and inhibits T cell function, resulting in the suppression of the immune system. Next, we present a comparison of cervical cancer and cohort samples that we analyzed in the same way. Some specimens are collected on a time course related to radiation treatment. PCA didn't show a good separation, but OPLSDA showed a significant separation of the two groups. In cervical cancer specimens, there was a significant increase in 65 metabolites and a significant decrease in 83 metabolites. A heat map of metabolites that showed significant changes in shown as well. This may have an effect on cervical cancer growth and survival. The decrease in PCs also confirms this result. Moreover, decreased levels of ornithine and beta-alanine and increases levels of carnitine and ceramide, which are characteristic ovarian cancer. Finally, the effects of radiation were compared. As a result, PC, methionine sulfone, proline hydroxide, citrulline, asparagine, and triglyceride decreased during radiotherapy and increased after radiotherapy. In contrast, carnitine decreased after radiotherapy. This suggests effect of radiation exposure. In addition, there are significant differences in some of the sphingomyelin, hexosylceramide, cholesterol ester, bile acid, and highly unsaturated fatty acids when compared with the therapeutic effect of radiation in PD group. These metabolites may affect the sensitivity and to radiotherapy in cervical cancers. In conclusion, we showed that the usefulness of metabolome analysis using KIT in large cohort studies and its application to biomarker discovery. Moreover, the metabolome analysis of patient's plasma may not only serve as a marker for cancer, but also be applied to determine treatment effects and predict prognosis. We will measure and validate more samples in the future. We also expect to measure the methods such as nucleic acid or proteome analysis to identify more reliable biomarkers. Thank you very much for your attention.